I'm very happy to be here, really, and to be dragged into this world. I am not even a brain biologist, so I'm a chemist and drug delivery person, but I'll do what I can to keep you interested. I don't know. Let's see what I can tell you. So practically, oh, here it is. So why I've been dragged to this field by Professor Rami Yaka here, because I have a very cool imaging system based on mass spectrometry imaging. And what is mass spectrometry imaging? You can think about it as a high resolution microscopy that, instead of looking at the anatomical features of the tissue, looks at distribution of molecules. Practically with my system right now, we can see almost any molecule below 1,500 Daltons of molecular weight. And in terms of brain studies, it gives you the variety of metabolites, drugs, neurotransmitters, and their precursors. And I think we can learn quite a lot of new information from this system. Just in a nutshell, how it works. It's based on mass spectrometer. This is a typical mass spectrum. We have a lot of peaks. Each peak is practically molecular ion. And well, so it's our raw data, how we translate it to imaging. There are several options. Maybe some of you are familiar with MALDI MSI. MALDI MSI uses laser on the tissue to desorb and ionize molecules, and then it gives a very nice images with high resolution. However, laser harms the tissue. So when you imaged your brain section, you're unable to do any other images because there are holes in the section. We like imaging system called desorption electrospray mass spectrometry imaging, or DESI-MSI. It is based on a solvent droplets, charged solvent droplets that we forward onto a tissue section. And these solvent droplets desorb and ionize molecules. It's done in open air, very simple, very fast. Not very, but like relatively fast. So this slide is kind of outdated, not kind of, it's very outdated. I made it 4 years ago, and I'm still using it just to show the difference. Nowadays, we are able to attain high spatial resolution between 10 and 40 micron spatial resolution. So we are able to differentiate very small features in the brain, and particularly in the reward system, as you will see with the project with Rami. So this is just like a small animation. You have a tissue. It's not a brain tissue, but like then you bombard it with your solvent of charged droplets. The splash of those droplets, you practically dissolve molecules from each and every place or place on the tissue, and then the splash of those droplets take the analytes into a mass spectrometer. Then you move your tissue in 2D in a very controlled manner, and then from each and every spot on the tissue you will get a mass spectrum, and then each and every such a peak can be translated, its intensity in 2D can be translated to a heat map. So at the end of this process, it's mostly untargeted process. We have also targeted instrument, but it's but just for high sensitivity. But for untargeted process, you will get, after one scan, you will get thousands. Oh, sorry. It's already Rami here. Thousands of maps, and then you can, find differences before and after treatment, before and after neurostimulant administration, and so on. So I really wanted to move into brain studies. And then Rami came and said, okay, we have, regions of reward system. And he came with the same notion as he mentioned today. The brain, after cocaine consumption, fails to utilize glucose properly when the person consumes cocaine, and even after they stopped consuming cocaine, still they cannot utilize glucose properly. And this is something that I can look at. I can look at the differences, metabolic differences of the brain before and after cocaine. So we decided in this experiment, in this study, we decided to go for acute cocaine administration and to look at the brains 24 hours after an acute cocaine administration. Why? Because we learned that the majority of effect of neuro damaging effects are happening within first 24 hours. And Samah Shaheen was involved in the study, in all the animal study here. So in my lab, Maria took this project. So Maria was a visiting student from Uppsala University. Now she's already PhD. So she came to my lab to study imaging, but she also brought a knowledge of microscale proteomics. So this knowledge was developed by her and her advisor, Professor Eric Janssen from Uppsala University. So they developed this workflow where they can do proteomics on very small samples, less than 20 microgram of proteins. So then we combined metabolomics, spatial metabolomics, with this microscale proteomics. So this is the workflow, practically like a lot of steps, but at the end you can get proteome from a very small sample and then compare it with metabolome, with spatial metabolome that we can get from my instruments, for our instruments. Okay, so so those are the brains after saline administration and cocaine administration after 24 hours. You can see a lot of difference in small molecules that we found, and practically we were able to construct the global effects of a cocaine. It's not an addiction yet, but it's neurodamaging effects. So indeed, we saw that glucolysis is very impaired. So we see a lot of lactate in all the areas of reward system. So if we look at TCA cycle, which is very important for mitochondrial, proper mitochondrial activity, so we see that the effects on different areas of the reward systems are different. So for instance, in prefrontal cortex and nucleus accumbens, you see a lot of aconitate elevation, whereas in VTA, aconitate is actually depleted. Then oxaloacetate goes up in VTA, so the effects are different. In the alpha-ketoglutarate and succinate, both of them are depleted. So here we know that the TCA cycle is probably impeded for VTA somewhere here, and for prefrontal cortex and nucleus accumbens somewhere here. Further, we saw that the enzymes by proteomics that are related to glutamate synthesis actually are induced. However, the glutamate itself, 24 hours after the administration of cocaine, glutamate goes down as well as GABA. However, here an aspartate goes down as well, and aspartate is eventually formed from glutamate. So, but here spermidine and taurine, they go up in the majority of regions. So it might be some kind of a mechanism by which mitochondria is trying to cope with oxidative stress that we see. Further, we see that a lot of that also citrate is not elevated, but here we have lipogenesis. So arachidonic acid, and I'll show you it here, is strongly elevated in all regions of the reward system. So are the lipids like phosphatidylethylamines, polyunsaturated phosphatidylethylamines that are synthesized in mitochondria are very much elevated, and it can also be a coping mechanism with oxidative stress that a mitochondria is facing. We also saw the induction in AMPA GLUR1 AMPA receptors. Probably is the first step of long-term potentiation that you heard about today, but we couldn't see further the effect of probably the start of addiction. Probably we will need further studies with longer exposure to cocaine, of course. So this was our first study, and really we found a lot of differences, and this all is published. But I really want to move to my second study, which is also funded by Israel Center for Addiction. So, and this study is currently done with Dr. Amit Lotan, who is the head of psychiatry department in Hadassah Hospital. And so Dr. Lotan is interested in brain diseases which are associated with high iron. And especially he is fond of iron chelation. And so one of his experiments included amphetamine induced hyperlocomotion, and he could reverse it by giving iron chelator before administrating iron chelator before administrated amphetamine, and he saw that hyperlocomotion was abolished. So, and probably because of amphetamine is elevating dopamine. So what happened was that the synthesis of dopamine was reduced before by iron chelation, because tyrosine hydroxylase is an iron-dependent enzyme, and so we could probably mitigate some of the effects of addictive neurostimulants. I was interested in this study, and I said, okay, let's take a look of what's going on in the brain of those mice. And I'm a pharmacist, so I'm interested in any therapeutic ability to mitigate any neurotransmitter disbalances, let's put it like that. So we definitely saw that when mice receive amphetamine, that the dopamine goes up in the brain. It was all done in Bregma +1. And when we administer the ferropron, iron chelator, before amphetamine, then this effect is abolished. The dopamine doesn't go up, and it's very significant. So next we were thinking, okay, is it truly because the iron is important to tyrosine hydroxylase, and this step is actually inhibited? However, when we looked at other things, so we looked at phenylalanine, and we saw that when we treat with diferapron, phenylalanine goes up, meaning that the step that diferapron actually inhibits by iron depletion is phenylalanine hydroxylase, which is also iron dependent and not tyrosine. Well, probably also tyrosine hydroxylase, but when we look at tyrosine, it's unchanged. So the main effect that we see is here. Then I said, okay, this is very nice. We can practically play with neurotransmitters by administering iron chelator. So, and as a pharmacist and drug delivery person, I said, okay, if we administrate, diferapron can be administered systemically, and then it has a lot of side effects. It has a granulocytosis. It crosses BBB, but you do not want to chelate all iron in periphery. So we decided, okay, we will develop an intranasal drug delivery system of diferapron. This was also done in my lab, so this is also one of my strengths, is to be able to develop drug delivery systems. So it's a delivery system that is based on FDA-approved components. So if needed, it can be moved into clinic. And so then we looked at the effect of intranasal diferapron on mice. Those are mice without amphetamine treatment. So we definitely saw diferapron in the brains after nasal delivery. Then we looked at dopamine, serotonin, and phenylalanine, and we saw that, as expected, dopamine went down. Serotonin also went down because it also synthesized by iron-dependent enzyme. Phenylalanine, we saw it before. It was inconclusive results here, but we saw clear effect of difereprone before. We also checked the levels of difereprone in the periphery, and so that after an intranasal delivery, non-surprisingly, we almost don't find any in the periphery compared to systemic administration. So this was good. So now we wanted to see how difereprone, our drug, is distributed between different bregmas in the brain and how it affects neurotransmitters there. So we saw a very nice distribution all over the brain, and we saw the non-surprisingly for you brain biologists, non-surprisingly, of course, the neurotransmitters were seen and affected, not in every bregma, but we definitely did this work and saw where, in which bregma, the dopamine, serotonin, and glutamate were affected. So it's, by the way, it's unpublished data, but we are kind of trying to gather more in order to publish it. Then we came to amphetamine studies. So we administered intranasal difereprone 2 hours before, 1.5 hours before administrating amphetamine, and then we sacrificed the mice half an hour after amphetamine administration, and we looked at the brain. We clearly saw that intranasal difereprone was able to abolish the effect of amphetamine on the brain in terms of dopamine. We are now doing behavioral studies for those mice. And now to cocaine. This is our current study. So cocaine also gives hyperlocomotion. Acute cocaine gives hyperlocomotion of mice in the open field, which can be rescued. It's not abolished, but rescued by systemic difereprone. So also we have some neurotransmitter studies of rescuing difereprone, rescuing cocaine effect with difereprone. So we saw that all neurotransmitters kind of were, the effect of cocaine was reversed. First of all, dopamine. We also saw it in glutamate. So this is a cocaine. This is cocaine with difereprone, and this is saline. So we see the rescuing effect in all the molecules studied. Also an interesting effect on citrate. So we will going to pursue the effect on also TCA cycle and other molecules. And I hope at some point we will move to a long-term cocaine here, and also in a collaboration with Rami. And we can do much more, so I'm open to collaborations, and I really want to be in this field of addiction. So I hope I convinced you that we can look at metabolome and probably microscale proteome of brain changes as a result of addictive substance administration, either acute or chronic. We can look at neurotransmitters and associated molecules. We can also track how, in a molecular level, neurotransmitters are changed. What has been, like in the case of phenylalanine and tyrosine. And also we can design nose-to-brain drug delivery if this is something that can be interested to maybe mitigation of addiction effects. Thank you all for listening. Thank you for inviting me here to give this talk, and thank you for collaborators in my lab.